Hi, I'm Katrin Preller. I'm super excited to moderate the panel on psilocybin neuroscience today. And together with Martin Matzen and Natasha Mason, we're going to explore what psilocybin does in the brain today. So I'm going to start first with my talk. I'll wait for another couple of seconds until everyone has joined. So my talk will be about psilocybin-induced time-dependent changes in global brain connectivity. And then after that, I'm very much looking forward to Martin's talk, and after that, Natasha's talk. And we'll be taking questions throughout, but we'll be answering all of them after all of the three speakers have completed their talks. And yeah, I'm very much looking forward to the session. So while people are still popping in, I'm going to share my screen and wait another few seconds to start with the presentation. So, nope. Just a second, please. Okay, so Martin, I can still see you. Does the presentation look okay to you?
Yeah, I can see it clearly.
Perfect. Okay. So good. So yeah, as I already said, I'm going to talk about psilocybin-induced time-dependent changes in global brain connectivity. And being the first talk of this session, I want to start off with a quick introduction into some of the methods we'll be talking about today in my talk, as well as the other two. So we'll be talking a lot about how psychedelics change how the brain communicates. And there are different ways of measuring this. And two of the measures that will pop up again and again during these talks is functional connectivity and effective connectivity. So functional connectivity basically measures statistical dependencies between regions of the brain. And there are various ways of how to measure these statistical correlations. Some of them are seed-based, where you just choose a seed and see how the rest of the brain is connected to this particular seed. And another one, which I'm going to talk about, is global brain connectivity, where we're looking at the correlation between each voxel, so each little department of the brain, with the rest of the brain. The advantage of functional connectivity is that it can give you a map of the whole brain, so you can see how each area of the brain is connected to at least either a seed or other areas of the brain. But it cannot give you anything about the causal relationship. So you cannot see which part of the brain influences which other part of the brain. For that to be able to do, we use a measure called effective connectivity or directed connectivity, where we can actually see how one brain area influences another. And one method that we have used in our studies with psychedelics is called DCM, dynamic causal modeling. And I'll start with that. So we use dynamic causal modeling to test one specific hypothesis about how psychedelics influence connectivity in the brain. And this hypothesis is called the thalamic filter model. And basically what it says is that a certain part of our brain, which is called the thalamus, and it's sitting here in the middle of the brain, is changing the way it filters the information to the cortex. And that is important because the information that we receive from the outside world, as well as from within ourselves, is usually filtered into important and non-important information. And the important information will pass through to the cortex, and the cortex will then have some kind of conscious perception of this information. And what Mark Geyer and Franz Vollenweider suggested is that this filter function is altered in psychedelic state in terms of having all information or a lot more information than usually pass through the thalamus, which then leads to sensory overload in the cortex, which is causing these psychedelic symptoms our participants experience. Now, as I said in the beginning, we cannot, at least for the time being, look at the whole brain with this method. So we had to pick 4 brain regions that are of particular interest for this model. So the thalamus in blue here, of course, the ventral striatum, which I'm not going to talk too much about because of time limitations today, 2 cortical areas, the posterior cingulate cortex, which is very much involved in how we process ourselves and information about ourselves, and another brain region, another cortical region, the temporal cortex. So we then gave placebo or LSD to our participants and looked at the changes that occur in the directed connectivity between these brain areas. And what we saw is that indeed the thalamus is sending more information to the posterior cingulate cortex, which is in line with the thalamic filter model. So the way it filters information is different. However, what we also saw is that it's not just sending more information to the whole cortex, but there are other brain areas like the temporal cortex, for example, where there's less information. So that means that it's not just a sensory overload as hypothesized in the model, but it's much more structured, and there are certain brain areas which receive more information from the thalamus. Now, of course, we also wanted to look at the rest of the brain, not just these 4 brain areas. And to do that, we used this method that I introduced in the beginning, a functional connectivity method called global brain connectivity. We did that with psilocybin, you can see the result here, as well as with LSD. And from just looking at this slide, without necessarily knowing too much about brain anatomy, you can already see that these 2 substances induce pretty similar effects in the brain. And what you can see here is that, first of all, we have a pattern of increased connectivity. And these are the areas which you can see here in warm colors. And it's striking how it is separated into brain areas which are responsible for processing our sensory information. So basically our visual system, our auditory system, our somatomotor system. So these brain areas which receive this sensory information, they're highly connected under both psilocybin as well as LSD. But then we also have these blue areas that you see here. And these blue areas are areas which are usually responsible for integrating all the sensory information that we receive, again, from outside as well as from within ourselves. And usually what these brain areas do, they bring all this information together so we can have a coherent perception of reality. Now, under psychedelics, these brain areas are loosely connected to the rest of the brain, which probably means that the way we integrate all this information that we receive is very different in the psychedelic state. We've also looked at how this changes across time. So the first scan we conducted here is after 20 minutes after administration. Then we conducted another scan 40 minutes after administration, and a third one 70 minutes after administration. And what you can see here is a mostly linear pattern of how these effects emerge. And it starts with increased connectivity in our visual system, which is very much in line with what our participants report. So usually the first effects they notice are changes in how they perceive, for example, the floor, so things start moving a little bit, etc. And then after 70 minutes, when we have usually the full-blown effects of a psychedelic state, this is where we see these changes or increases in brain connectivity in our sensory brain regions, as well as the decreases in our association regions. Another interesting thing that we found in this study was that the way our brain is wired is connected in the baseline state or under placebo is very much correlated with the changes that are induced afterwards. Again, this is increasing over time. And the reason why I think this is important is because that might mean that by scanning people in their baseline, in their usual state, might already give us a hint of how they might react later on to the psychedelic. And especially when we're talking about the framework of psychedelic assisted therapy, this might give us a measure to stratify patients early on, whether they will have a good experience or a bad experience or a strong experience or a weak experience. Of course, the study was done in 23 participants, so it's a very, very small study, and this has to be kept in mind, especially when we're talking about correlation analysis. But it's a first hint that, you know, the way our brain is wired in each individual might influence how we then react to a psychedelic compound. So to sum all this information up really briefly, is what we've seen in our studies is, first of all, that the thalamus is sending information differently to the cortex. And we've seen that a lot of information is, or that at least there's a stronger connectivity between the thalamus and sensory brain regions. And the sensory brain regions, again, are highly connected with the rest of the brain. Whereas at the same time, the brain regions which are responsible for integrating all this information are connected loosely to the rest of the brain. So what that means is we probably integrate the information about ourselves and the rest of the world differently, which could explain why people experience these visual alterations, but it could also explain why it might help people to kind of look at them in a different way, to look at the world in a different way, and break out of these ruminative or negative thinking patterns that we see so often in, for example, depressed individuals or other patients with psychiatric disorders. So to sum this up really briefly, we have seen alterations in the thalamic gating that I described, and at the same time, the higher integration of sensory into experiences and the disintegration of associative brain regions. We've seen that the baseline connectivity may represent a predictive marker, which may be really important in the framework of personalized medicine. And the other thing that we definitely need is more rigorous scientific studies to really understand the neurobiology of psychedelics in the human brain, and also to capture their full clinical potential. And two of these rigorous scientific studies will now be presented by first Martin and Natasha afterwards. So thank you so much, and I'll hand over to Martin.
Thank you, Katrin, for a great presentation. And I will just try and share with you my screen. Try and see how that goes. Share. Did this work? Can you see my screen? Good. Yes. Good. So thank you, Katrin, for the introduction, the great presentation. So I would like to share with you some really exciting findings that we have produced over the last 4 years here in Copenhagen. And so psilocybin, all you know this compound probably, so psilocybin is the main psychoactive constituent of magic mushrooms. And really psilocybin is a prodrug. So this phosphate moiety right here is cleaved off by phosphatases in the blood and in the gut. And this yields psilocin. And psilocin is actually the active compound acting in the human body after intake. And we know that psilocin is important for psilocybin effects. And we showed this in a PET study we published last year. And here we showed that psilocybin binds to the serotonin 2A receptor in the cortex to a high degree. And these three figures summarizes our main findings. So in the figure here to the left, you see that the more psilocin you have in the blood, the more psilocin you have bound to the receptors, that stimulates the receptors. And the figure in the middle here shows that serotonin 2A receptor occupancy is positively correlated with how intense the experience is. So the more psilocin you have stimulating those 2A receptors in the brain, the more intense the effects are experienced. In the figure here to the right, you see that the more psilocin you have in the blood, the more intense the effects are. So these three figures really show how that the more drug you have in the blood, the more you stimulate the receptors and the more intense the effects they are perceived. This is a figure from a landmark study by Carl Harris and colleagues from 2012, and this shows how two backbone structures of the default mode network, they're desynchronized. So in the red, you have the medial prefrontal cortex, the MPFC right here. In the blue, you have the posterior cingulate cortex. And this shows that after IV psilocybin, these two key structures of the DMN, they get desynchronized. And the DMN is an interesting network because it's been implicated in ego and in having a sense of self. A reanalysis of the same data, the same group showed that in general, there was an increase in between network connectivity after psilocybin. So the networks became less segregated than they were in the non-psychedelic state. So these studies, with the exception of Kathrine's really interesting study, have been snapshots. So you give a drug and you take one picture of what happens in the brain. But really the psilocybin experience unfolds over time dynamically for about 5 to 6 hours. And the same goes for psilocin levels, we know that, and also for the intensity. And so really in order to capture this, you need a bit more elaborate setup than just taking a single picture of the brain. So what we, and that we tried to do. So what we did here was that we included 15 healthy participants with limited or no experience with psychedelic drugs. They were young adults, and they came in on a day, and then we put them in an MRI scanner. We did this before they were given drug, and then after they were given drug. Immediately after we took some pictures of the brain using bold fMRI, we asked them how intense the experience was on a scale from 0 to 10. And after that, we took a blood sample so we could measure the psilocybin levels, or the psilocin levels in the blood, immediately connected to the imaging acquisition and this intensity rating. And we gave a psilocybin dose that was 0.2 milligrams per kilogram, which is a moderate to high dose, and then 0.3 for the remaining 11 participants, which is the highest dose that has been administered in the MR so far. We did 4 post-drug acquisitions of this data. Our hypotheses were that psilocin, plasma psilocin level, PPL, and subjective drug intensity, SDI, would correlate negatively with connectivity within the DMN and in individual networks. We also hypothesized that psilocin and intensity would correlate positively with between network connectivity. And this would reflect that there was reduced segregation of these functional networks. So what did we see? Well, we saw that as a function of time, psilocin levels, they rose and they fell again, and this is unsurprising. We saw the same thing for intensity levels. You might notice here this subject in pink was a bit strange. He rated the intensity to be 0 for almost 3 hours, after which he finally broke through and had a psychedelic experience. And I think this is partly explained by the slow pharmacokinetics exhibited by this subject. So the onset was really gradual and slow for him. When we correlate psilocin levels and subjective drug intensity ratings, we see that there's a positive correlation, which you would expect, and it's consistent with the results I showed you previously. We observed that psilocin was negatively correlated with connectivity within the DMN, and the same we observed for intensity. So the more psilocin you have in the blood stimulating those 2A receptors in the brain, the more intense the effects are perceived, the more the DMN is desynchronized. And this is a video showing the effects. You can see that the MPFC gets decoupled with the PCC as time passes, as psilocin levels, they rise and reach a maximum at roughly 2 hours after psilocin has been ingested. And this really shows how this method can capture some of these neuro imaging and pharmacological dynamics, how they change over time. Yes. We also saw that psilocin and intensity correlated positively with between network connectivity, such that the more psilocin you have in the blood, the more intense the effects they are experienced, the less segregated these functional networks they appear to be in the brain. We also looked at individual networks. What was the effect of psilocin and intensity on the connectivity within and between networks? And first of all, I think one thing to notice is that these effects weren't randomly distributed. When you look at how these networks, they are spatially located in the brain, you see that there's a certain pattern to it. You have the default mode network at one end. You have some higher order networks such as salience, dorsal attention, and executive control networks in between. And then on the other end, you have sensory networks and unimodal sensory networks such as auditory, visual, and sensory motor networks. And what you see here is that the DMN was desynchronized, the same thing for salience network, but the DMN increases connectivity with the salience network, with the executive control network, and with the dorsal attention network. And actually the dorsal attention network and executive control networks, they synchronize increasingly with the visual system, auditory and sensory motor systems as a function of psilocin and as a function of intensity. We also looked at some whole brain changes induced by psilocin and intensity. And first we looked at a measure called local correlation, or LCOR. And what is LCOR? LCOR measures how synchronized two voxels are, or not two voxels, but local voxels is. So it measures how disturbed or coherent the signal is going to be as a function of psilocin and intensity. And what we saw was that it was in the key regions of the default mode network, in MPFC and PCC, also in visual cortex and the dorsolateral areas belonging to the executive control networks, that psilocin and intensity, they induced a dose-dependent reduction in local correlation. And we interpret this as a reduced integrity of these networks locally. We also employed a similar measure to what Katrin has showed. This is called a G-core. So Katrin has used some different methodology than we have, and this might affect how our results they compare. But what we observed is that as a function of psilocin and intensity, you see an increased connectivity between voxels located in the dorsal attention network, in the executive control network, with the rest of the brain. And we interpret this that there's a reduced segregation of the DAN and the ECN with the rest of the brain. Doing a study like this, where we map over time psilocin levels, intensity levels, and what happens with connectivity patterns in the brain, allows us to produce what I a bit cheekily could call a map of the trip. So here you see that for what we assume is going to be the average plasma levels of a 25 milligram psilocybin dose, you see psilocin levels rise and fall. You see that intensity levels, they reach a plateau and fall again. And you see that connectivity within the DMN is reduced and then starts to go back to baseline as drug is washed out of the body. And you see a reduction in within network connectivity and an increase in between network connectivity, reflecting reduced network segregation. So in conclusion, we observed that psilocin levels and intensity, they correlate with connectivity within and between networks, that the integrity of the DMN, executive control networks, and the salience networks are reduced, and that neighborhood networks, they synchronize, they desegregate. It's not randomly distributed in the brain, but it seems to be a spatial pattern to these effects. We see that in the global analysis that the dorsal attention network and the executive control network, they desegregate with what you can call internal and external networks. And I think this shows quite clearly that psilocin is absolutely crucial for the effects of psilocybin, and that measuring psilocin in the blood perhaps could account for individual differences in treatment response in such studies as depression. So I would like to thank you for your attention and thank the whole team at the Neurobiology Research Unit for helping out with this study. Thanks.
Great. Thank you, Martin, so much. Thank you for this really great talk and sharing these super interesting results. We'll move on to Natasha now, who I believe has pre-recorded her talk due to a slow internet connection, but she will be available for the Q&A afterwards.
Natasha Mason. I'm a PhD student at the Department of Psychopharmacology in Maastricht University. And today I will be sharing with you a recently completed study assessing the acute effects of psilocybin on glutamate concentration levels and the relationship between functional connectivity and subjective state. And before I begin, I just want to say that this was the first psychedelic conference that I ever attended back in 2016. So I'm extra excited to be able to come back to the next one and share these exciting findings with you all. So by now we know what psilocybin is. We know that as a psychedelic, once ingested, it induces a profoundly altered state of consciousness, including things like changes in perception, like visual perception. So this is closed eye, open eye alterations, as you can see in the example here. Enhanced emotional state, which can range from positive bliss to negative fear. Widespread changes in cognition and thought content. And arguably a more unique effect of psychedelics are changes in the self-experience. So I'm assuming most people at this conference are familiar with the term ego dissolution, this loss of sense of self, dissolution of the boundary between yourself and your environment. Oftentimes when people experience this, they report back that they felt one with everything. I also think right now we know why we are interested in substances like psilocybin. So it's because recent clinical studies suggest that psychedelics can provide fast-acting, long-term symptomatic relief for individuals suffering from disorders like depression and anxiety. Thus, this raises the possibility that research into these substances may identify novel therapeutic mechanisms for these very difficult to treat, very prominent disorders. So an important question is how these substances work in the brain to elicit such effects. So it's been established that classic psychedelics act on 5-HT2A receptors, which is a suggested primary mechanism of action for their hallucinogenic effects. However, we know that activation of these receptors is not the entire story when getting from here to here. So as this figure illustrates, stimulation has another downstream consequence, which is an increase in extracellular glutamate levels in the cortex. So why are we interested in this downstream effect? That's because preclinical studies have implicated this enhancement of cortical glutamate in acute psychedelic-induced effects on brain function and behavior. And it's been suggested that this release of glutamate subsequently increases levels of BDNF, so a protein involved in neuronal growth and survival. And thus it's been hypothesized that glutamate-induced neuroplasticity may be underlying some of the persisting effects that we see in clinical trials. So all this evidence that you see on the screen emphasize the role of glutamate in the acute and persisting effects of the drug in preclinical studies, but the effects in humans have not been assessed. So that's what we're interested to do. Look at the effect, the acute influence of psilocybin on glutamate levels in key brain areas implicated in the psychedelic experience, so the medial prefrontal cortex and the hippocampus. And as glutamate has been implicated in the psychedelic-induced effects on behavior and brain function, we wanted to look at the relationship between glutamate levels and what I have termed here key features of the psychedelic state. So this includes the experience of ego dissolution, this acute loss of sense of self, which has actually been found to correlate with long-term outcomes like increases in well-being. So the more ego dissolution acutely, the more positively experience ego dissolution acutely, the better people feel in the long term. And in regards to brain function, this includes network connectivity. So it's been repeatedly found that these drugs decrease connectivity in several large-scale resting state networks. Here, a lot of the focus has been on the D-FOMO network in particular, and studies have found that decoupling within this network is associated with the experience of psychedelic-induced ego dissolution. So as the medial prefrontal cortex and the hippocampus anatomically overlap with the DMN, it's a good starting point to assess the role of glutamates in both. So to do this, we implemented a standard study design, double-blind, placebo-controlled. 30 individuals received a medium dose of psilocybin, 30 received placebo. And after they received their treatments, about 60 minutes later, they went into our MRI scanner. So here we performed magnetic resonance spectroscopy in order to quantify glutamates in our brain regions of interest. And then we also collected resting state data so that we could look at within network functional connectivity. And for this, we just repeated the analyses that previous labs had done with these substances to replicate it and see if we found the same results. In regards to ego dissolution, we use 2 validated questionnaires. So the 5-DASC, which I'm sure you'll hear a lot in this conference, and the ego dissolution inventory. And what I want to say about the experience of ego dissolution is that it can be both positive and negative. So we quantified, or we looked at these kind of separately in the end. So in the 5-DASC, we have this term called oceanic boundlessness. So this feeling of one with the surroundings. This is more positively experienced ego dissolution. So we see that in the 5-DASC and ego dissolution inventory. But there's also a kind of fear that can come with losing your sense of self, losing control. And that's termed negatively experienced ego dissolution. And we can quantify that with this anxious ego dissolution dimension in the 5-DASC as well. So what did we find? Well, unsurprisingly, we found that individuals under psilocybin had higher ratings of both positively and negatively experienced ego dissolution than those under placebo. More interestingly, when we look at glutamate, so these are rain cloud plots. On the far right, you see the dots. These are individual data points. In the middle are the box plots, and on the left are the distribution of the data. And here we see that under psilocybin, individuals had higher levels of medial prefrontal cortex glutamate compared to placebo. So as one would expect. In hippocampus, though, we see the opposite. We see under psilocybin, individuals have lower levels of glutamate compared to placebo. In regards to within network connectivity, so we did a full brain analysis. This is to show you if you're interested, but for the sake of this presentation, we find, just like other studies did, that psilocybin reduced within network connectivity under psilocybin compared to placebo in the default mode network. And then assessing the relationship between brain and behavior, we use a canonical correlation analysis. So here we use the 4 biological predictors. So this would be glutamates in our brain areas of interest and our 2 components of the DMN in order to predict ratings of both positively and negatively experienced ego dissolution. And we found when we did this that function 1 showed that the dominant glutamate concentrations in the medial prefrontal cortex were the dominant predictor for the dimension anxious ego dissolution. So the more glutamate in the prefrontal cortex after psilocybin, the higher ratings of anxious ego dissolution. As for function 2, we find that the less glutamate in the hippocampus after psilocybin, the more ratings of positively experienced ego dissolution. So in summary, we find this region-dependent alterations in glutamate. In the medial prefrontal cortex, there's higher levels after psilocybin compared to placebo, whereas in hippocampus there are lower levels. And as I said, there's a region and domain-specific relationship with subjective effects. So the more glutamate in the medial prefrontal cortex, the higher ratings of negatively experienced ego dissolution, the less hippocampal glutamate, the higher ratings of positively experienced ego dissolution. So in regards to what this means in the grand scheme of things, so studies like this provide further information about both the acute action of these substances and also provide potential mechanisms of persisting effects. Now, I would love to talk another 10 minutes about this and discuss this with you. Don't have time, of course. I will say that we recently published this data in neuropsychopharmacology. So if you are interested, there's a full discussion there, but I'm also obviously very happy to answer any questions after this presentation or to discuss with you throughout the conference. So with that, I'd like to say thank you for your attention and of course, thank everybody involved in this study.
Natasha, thank you so much. Thank you so much for this great talk. We have a couple of questions in the Q&A section, and I think we have another 10 minutes to quickly go through them. And I'm just going to start asking the first question to Martin, and this is: which atlas parcellation did you use for your network analysis?
Yeah, that's a good question. So you always need to make a decision of which atlas to use when you do an analysis like this. And we chose a not such often used atlas, which is a delineation of 36 regions that's been published by Marcus Rakel in 2011. But if you look at how these regions are distributed, they match pretty well the atlas published by Thomas Yeo in 2011, even though there's some discrepancy to it. So that's the quick answer. The Restless Brain by Rakel in 2011, you can see the coordinates for the ROIs we used here. But it's only 36 regions, and the extent to which you would observe other effects using a different population than what we did, I'm not so sure of, but something that might be interesting to explore. There's several atlases out there, and that was a big part of, there was a big discussion here, which atlas to use.
Wonderful, thank you so much, Martin. I'm going to answer the next question here, which is, how do you consider top-down versus bottom-up effects in your model? And that, I think, is a really, really interesting question. So for the data that we have right now, especially the ones where we looked at connectivity from the thalamus to the cortex, it points towards increased bottom-up effects. But given that the brain is so much more complicated than most of our models can actually capture and most of our analysis can capture, I'm sure that there is something, you know, that there are certain top-down influences or changes in top-down processing going on as well. But for, you know, the data I presented so far empirically, we have shown that there is a certain degree of increased bottom-up processing. Okay, which brings me to the next question to Martin. What is the connection between salience network and DMS?
I need to have explained what DMS stands for.
Okay, maybe Giorgio, who posted this, can clarify in the Q&A right now. Or we'll just see if he clarifies. If he does, we'll take that question again later. I think the next question is going to Natasha. Did ego dissolution effects correlate with changes in self-concept, and how?
Yes, I think I also need a definition, though, is what they mean by self-concept in this. Okay. So, I will say that in regards to the correlations with ego dissolution and any effects, we were only interested in looking at the relationship between glutamates and within network connectivity of the default network and ratings of ego dissolution. So we didn't look between the different dimensions, for example, on the 5-DAS.
Okay. There is another question for you here, and so again, I'm reading this in real time. If increased glutamate in the hippocampus causes a higher frequency of positive ego dissolution, does this indicate, does this indicate some individuals are more likely to have that experience based on their neurobiology? Or is it the resting glutamate levels in medial prefrontal cortex versus hippocampus caused through other factors? Do you have any plans to study this?
So does this indicate some individuals are more likely to have experiences based on their neurobiology? I mean, I think that's a fascinating question, and I think that's also what you were looking at as well, with this baseline connectivity kind of predicting subsequent response. I would, off the top of my head, say yes, of course. People will respond differently to any drug you give them based on their underlying baseline state. Do we have plans to study that in the future? Personally, not. I do not, but yeah, in regards to personalized medicine and predicting responses, I definitely think that's an important thing to look at.
Thank you. And another question for you. Was there a significant difference in intensity between the positive and negative experiences? Could that difference in intensity explain some of the effects?
A significant difference in intensity. I mean, people did not rate extremely high levels of anxious ego dissolution, for example. The higher levels were definitely in the positively experienced ego dissolution. Again, I didn't only say significantly, I didn't statistically test this, but looking at the ratings that we had. So there are differences there, as other studies see as well. Usually anxiety in healthy individuals who have been prepped for this experience is quite low. Could that difference in intensity explain some of the effects? Yeah, I guess I don't know the answer to that. Yes, of course it could, but yeah. We did find these correlations. We found these predictions. We were able to predict responses based on glutamate concentration levels. So I think that alone is quite interesting.
Thank you. Martin, could you briefly repeat the name of the atlas again? It seems like some people were not able to catch it due to a bad connection.
Yeah, so if you look up Marcus Raichel's 2011 paper called The Restless Brain, you see the 36 regions or coordinates for the regions that we have used in our analysis.
Thank you. And Natasha, I was wondering, you showed these region-specific effects of glutamate in after administration of psilocybin. So how, I mean, you make this link between glutamate and neuroplasticity, of course. So what is your interpretation of this region-specific effects on neuroplasticity? Because so far people have mainly been talking about, you know, psychedelics increase neuroplasticity, and in a way you'd expect that to occur across the whole brain. Now you're showing that, well, actually the story might be so much more complicated. So what is your take on that? What is your interpretation?
Yeah, I think that's a really good question. And this really demonstrates that these substances do have different effects in different regions, you know, depending on the receptor population there and probably the proportion of the receptors. I mean, this would suggest that if psychedelics do induce neuroplasticity dependent on this increase in glutamate, which are already many assumptions, that this would be taking place in the medial prefrontal cortex and not the hippocampus. And I think there have also been previous rodent studies not directly assessing glutamate or neuroplasticity, but have found, for example, reductions in BDNF microRNA in the hippocampus as well. So yeah, this would just suggest that it's not a net brain effect, right? So it would be region dependent.
And I think, Martin, there is a clarification on the DMS, which was supposed to be default mode network.
Yeah, thanks. I looked at the letters. S was a bit far from N on the keyboard, so it's nice to be that clarified. So there's a theory that the salience network, what it does is really set of regions, including the and prominently dorsal anterior cingulate and the anterior insulae, that what these set of regions do is that they map how important stimuli, either internally derived from the DMN or externally derived, that comes in through some of the attentional networks, how important that is. Like that is where to shift attention to, whether you should think about something, an essay you should hand in tomorrow, or whether you should be wary of the car that comes towards you. So they're not directly related. I think the DMN and the salience network, as so far as I think about them, but more you can envision them as sort of discrete events that take place different times in space. And presumably it's because you have the serotonin 2 receptors also in the salience network. So when you stimulate those receptors, you see like a desynchronization within the networks.
Thank you, Martin, so much. I realize we are at time. We have answered most of the questions, but unfortunately not all of them. But if I understand correctly, there's going to be an after talk, so maybe we can discuss the still open questions there then. Thank you so much for joining the session. It was so much fun, and enjoy the rest of the conference.
So thank you all. Before we stop this stream, I want to announce a couple of things. So we have a breathwork session now for the people that are interested. And we have 3 other sessions coming up in around 25 minutes. And those sessions will be in the Hofmann Hall. It's about different modalities of psychotherapy with psychedelics. The other one is Wade Davis, and the other one is it's actually, it's ethics and transparency in psychedelic research. And finally, we still have posters in the WUFA app that we really would like people to engage with. So to see which is your favorite poster, the top posters will receive a prize. So please have a look at them. And also, as Catherine just mentioned, there is an after talk in the WUFA app if the speakers are available for that. And you can find it here in the chat, and also on WUFA there will be a link for that in the WUFA channel for this talk. So thank you very much all, and see you all soon.
Thank you.